All right, welcome back. This is the final and part 5 of the materials to help you understand the testing of bacteria as far as clinical laboratories are concerned. And I'm going to focus on the biochemical tests. And what that means, of course, is the biochemical reactions, certain tests like catalase, oxidase, urease. These sorts of things are enzymes that are produced by bacteria that do something, but usually are unique to a particular species and genera. And that really helps in the identification. A lot of these tests are really just simple. In addition, of course, like you see in the slide, this is a very snotty plate. It shows bacteria producing a very thick polysaccharide, which is really nasty. It's most likely a Klebsiella. I don't know what that one particularly is, but they kind of identify themselves by the enzymes they make. So here are the tests: urease, catalase, oxidase, the Volks-Progner test, the VP, methyl red, Volks-Progner, coagulase, and motility auger. Now these are really very simple. The urease test is to identify bacteria that's capable of hydrolyzing urea. That's why they call it the urease test. And Proteus vulgaris and other types of enterics will do this. It forms from the urea a weak base ammonia, and that raises the pH above 8.4, and we use phenol red as an indicator, and it turns it from yellow to this sort of a pinkish color that you see here. It's really very pronounced, so we go from yellow to pink. Proteus mirabilis is a rapid hydrolyzer of urea, as you see, and the tube on the far right is inoculated urease negative organism, so you can see that, and on the far left is one that's uninoculated. So if it's a true negative, you can see that it turns as yellow and this hot pink, I guess you could say, and you can tell if it's been uninoculated because it has this sort of orange color. So that's the urease test. It's really simple, and it raises the pH because of the ammonia, which is a weak base, and it turns it this Hello Kitty pink. Yeah, I said that. So the Catalase test is really easy, and I used to carry a bottle of hydrogen peroxide in my pocket, and I would just drop it on a culture, and it lets me know right away if I'm dealing with a staph or a strep. That's usually the hardest thing in a lab, a diagnostic lab, and it gives me kind of a quick response. It does immediately. You start to see the foaming of if it's a catalase positive, as you can see here. And again, it's the staph that will do this, also micrococcus, but at least it sends me off in a direction that I'll start looking at staph, Simmons citrate and things like that. Obviously I'm dealing with a gram-positive, and then I can tell right away that streptococcus and enterococcus tend to be catalase negative. So that's the catalase test. We're almost there. The oxidase test is really easy as well. It's usually the chemical is in a little breakable tube thing that's in a plastic tube, and inside is a glass, a very easy to break glass container, and so what happens, it's commonly used to distinguish between oxidase negative Enterobacteriaceae or oxidase positive Pseudomonas. And so you can see there's a positive test is that really dark purple, and a negative test is kind of unremarkable. It just kind of doesn't have any color. So the cytochrome oxidase transfers electrons from the electron transport chain to oxygen, the final acceptor, and reduces it to water. The oxidase test is an artificial electron donor, and the acceptors are provided when the electron donor is oxidized by the cytochrome oxidase, and it turns it dark purple, and that's a positive test. And so that one of the things that we can do is to help identify Pseudomonas aeruginosa, because it'll turn that oxidase positive purple, as you see there. So it's a simple test. It's just one more thing that we can use to identify making the preponderance of the evidence if we're after Pseudomonas. The methyl red Volks-Progner It's used to determine fermentation pathways that utilize glucose. And in a mixed fermentation pathway, glucose is fermented and produces several organic acids: lactic, acetic, succinic, and formic acids. And the stable production of enough acid to overcome the phosphate buffer results in the pH lower than 4.4. The pH indicator, which is just methyl red, is added to the aliquot in the culture broth below, and the red color will appear first. And then if the methyl red turns yellow, the pH is above 6, and you start to see mixed acid fermentation. And so the first tube on the right, 2,3-butanediol fermentation pathway will ferment glucose and produce 2,3-butanediol end product instead of organic acids. And in order to test this pathway, an aliquot of the Volks Progner culture is removed, and naphthol and potassium hydroxide are added, shaken, and set aside for about an hour. And the Volks Progner test detects the presence of acetoin and a precursor of 2,3-butadiol. Now that's a lot of stuff. It's just a really easy test, and if it's positive, as you see here, then it turns red, then you know. If the culture is negative for acetoin, it will turn a brownish green to yellow, and as you see over here on the left, the yellow. So Enterobacter aerogenes and Klebsiella pneumoniae are MR negative and Volks Progner positive. Pseudomonas aeruginosa is glucose non-fermenter, that it's negative for both. So you would see it as yellow. So Enterobacter aerogenes would be positive as well, and Klebsiella pneumoniae. Pseudomonas aeruginosa is negative, so that's a good way to determine or distinguish between those two. Simple test. And the Coagulase test is an enzyme that clots blood plasma. That's all it does. It's an enzyme, ASE, and it coagulates blood plasma. The test is performed on gram-positive, catalase-positive species to identify Staph aureus. Coagulase is the virulence factor of Staph aureus, so it tends to cause a clotting of a factor, so it seems to prevent your immune system from getting in there. Formation of a clot around the infection causes bacteria protects it from phagocytosis. This test differentiates Staph aureus from other negative species. Coagulation negative, so Staph epidermidis comes to mind as a negative. You can see the liquid part, and then the aureus clumping. That's the Coagulase test. Just a few more. Motility auger is really easy. This is a non-modal. I'd stab it with a stabbing loop. It's usually a standard loop without the end on it. The looping in the end is just a stab, or you can use it with a loop, and it seeds the bacteria in there. If it's modal, it starts to move out, and you can see that it's just growing all the way through the tube. And if it's turbid, then it's mostly the bacteria moved away from the stab mark. If it's non-modal, it just sits there. And that's a really easy test, so you can determine if it's modal or not. And that's all I have for you. That was an easy one. Those are the simple tests that we run to help us identify and convict in court with the preponderance of the evidence. It's just more things that we can use to identify. So I hope this series has been helpful, and the next section 6 will be the actual assignment and the choosing of the sort of a tree, of the dichotomous tree of how to identify bacteria, and I'll go through that in that section.